Hi everyone, welcome to the webinar, and my name is Manvi Sharma, and I am a postdoc with the High Altitude Program at Nature Conservation Program, and I'm very happy to be here to introduce Munib today. Dr. Munib Khanyari is a fellow postdoc and he's a program manager at the Nature Conservation Foundation in Bangalore in India. And Munib before this, he did his PhD from University of Oxford and University of Bristol. And before that, he did his master's from McGill University and bachelor's from McGill University. And he has very varied research interests. He's interested in studying disease ecology, population ecology of ungulates, and also he's led many interesting discussions on ethics of conservation and conservation practice. So I'm very excited to welcome him here today. And he'll be talking to us about parasite transmission in the Trans-Himalayan landscapes. And welcome, Munib.
Hey, thanks so much, Manvi. It's always a pleasure to be introduced by a fellow friend and colleague. And I'm really excited to have everyone here today. Thanks for joining in. I see in the chat people are from different parts. Somebody's even joining from Texas I'm not even sure what crazy time it is. So thank you. So basically, as Manvi said, I'll be talking to you guys a little bit about a paper that I published during my PhD where we looked at parasite transmission in a Trans-Himalayan system, and we'll have a discussion around that at the end. So I'll, without further ado, I'll just sort of share my screen. So yeah, basically, I'm going to be talking to you about how we try to account for multiple hosts and climatic effects in a remote landscape, in a remote multi-use landscapes on parasite transmission and their control. I mean, this is a bit of a mouthful for a title, but hopefully it'll make sense as we go along the talk. As I said, this is going to be a case study from the Indian Trans-Himalayas. This is what you see in the picture. It's from the study site, which I'll describe in a little bit. And a lot of our talk will be revolving around these ungulates, the blue sheep that are primary prey to the snow leopard. And, I just wanted to use this picture to show that while these are really high remote areas, they're also multi-use landscapes, as you can see with the background in this context being that of agriculture. Just before I start, I want to say, as Manvi alluded to, this was part of my PhD with the Interdisciplinary Center for Conservation Science at the University of Oxford and the University of Bristol, and also collaboration with two NGOs that I'm now currently working with, the Nature Conservation Foundation, which is the India country partner to the Snow Leopard Trust. So probably to a lot of you, this is a very familiar map. Essentially, what I'm trying to show here is this this dark lit area within Central and South Asia is what we term as High Asia. This is the landscape where you have the mountains of Central and South Asia and their interconnected plateaus, high plateaus, especially in high plains. And within this dark landscape, you have these different mountain ranges that are colored in different colors that essentially forms the global range of the snow leopards. As you see, it traverses 12 countries all the way down from Bhutan, Nepal, and India, all the way up north to Russia and Mongolia. And it has all these big mountain ranges such as the Himalayas, the Tibetan Plateau, Tian Shan Mountains. So just to give you a context of spatially where we are located in terms of the larger snow leopard landscape and their prey. And within this large area, a lot of my personal work has concentrated in this area in India, which is the sort of the northwestern Himalayan region of Himachal Pradesh, Ladakh, and now even Jammu and Kashmir. So that's where we are going to be geographically. A lot of you who work in these landscapes across Central and South Asia would know that a lot of times these are beautiful remote landscapes, very harsh winters, home to the predator snow leopard, their primary prey, in our case, the blue sheep, but in many other areas, we have things like the ibex or different mountain ungulates, markhor, argali, so on and so forth. But very importantly, even though these are remote landscapes, far from major cities often, they're also home to a lot of different people, predominantly pastoralists or agro-pastoralists, that at least in part depend on livestock for their livelihoods. Just to give you a context of where we're situated in India, like if you zoom in from the map that I showed a couple of slides ago, we are situated, a lot of the work, at least in this talk and our program, is situated in Himachal Pradesh, which is this northern Indian state. And within Himachal Pradesh, about 20,000 square kilometers exists of potential snow leopard habitat. And that's where we work at large, although we have sites where we do a lot more on-ground sort of long-term work, of which one I'll be talking about just in a bit. Before I get into the whole parasite aspect, the disease aspect, I would really wanted to set some context to the whole sort of work. This is a map from a paper that Manvi, in fact, led and some of my colleagues led. All I wanted to show from this was up in A, you see the state of Himachal Pradesh. And what we did there was to get a sense of snow leopard occupancy across this landscape. And I'm just going to speak very quickly about this because this is not the crux of the talk today. What we realized was that 20,000 square kilometer within Himachal Pradesh essentially can be divided into 3 strata. One of high snow leopard occupancy, which is this yellow range, one of low occupancy, which is sort of this light green range, and some areas which potentially were snow leopard habitat, but we didn't know much about. And then when we went ahead and studied these areas more in depth to understand how snow leopards and their prey, are doing in these landscapes. So we representatively sampled these areas. We had 3 areas where we camera trapped and did some ungulate surveys in the low areas, 4 sites in the high areas, and a few sites in the unknown areas. And this was done on ground, covering roughly 5,000 square kilometers. And what we found, and this is something I want you guys to keep at the back of your mind as you listen to me, is we find this somewhat linear relationship between prey densities and snow leopard densities. In other words, it's very, very important for enough prey to be there for snow leopards to exist in these landscapes. Obviously, there's a lot more factors, but this, although somewhat intuitive, was important to sort of set the context in these areas. So prey is quite important, as we see from this large-scale survey. Another quick point of context, I mean, for all of you who know me, know that I love mountain ungulates. So I always find ways to think of their importance, but they are quite important regardless of me having a bias towards them. And I term them to be the sort of ecological fulcrum of these Trans-Himalayan landscapes. So imagine, if the Trans-Himalayas was the seesaw that can tip up and down. I sort of tend to believe that at the center of this lie the mountain ungulates, the prey species to the snow leopard, and the primary herbivores of the system. And there's enough studies to show that, the grazing that is done by these mountain ungulates in these systems particularly has a positive impact on the vegetation of this system. Everything from nutrient cycling to, impacting vegetation composition of these areas. So, these primary grazers are very, very important for the primary producers in these low resource systems. At the same time, as shown by my previous slide and some other studies as well, these prey species, in my case, the blue sheep and the ibex particularly, are super important for predators like snow leopard in these ecosystems. In fact, they are direct sort of determinants of their population as well, even though, or even in landscapes where livestock exists. But interestingly, as I was saying, most of these areas, not only in my field site, but across Central and South Asia, are human-use landscapes where, rightfully so, people have been living for millennia, particularly grazing their livestock or even doing agriculture. So livestock, particularly small-bodied livestock, which has very similar ecological roles to these their wild cousins, the caprids, sort of, I tend to believe, fulfill a very similar ecological role potentially to the wild ungulates in these systems. And there are studies to show that snow leopards might depredate or eat livestock and are livestock might be important to snow leopards' diet, even though they might not sustain themselves entirely on livestock. And at the same time, there's also a lot of studies to show how livestock might be both important and detrimental to the primary production in these remote and low resource landscapes. It might lead to overgrazing and things like that. So just to sort of use this slide to give a sense of the different and nuanced interactions, particularly ecological interactions happening in these landscapes. I tend to focus a lot, or at least my PhD tended to focus a lot on this side of the interaction, which was a lot to do with the wild ungulates, the domestic ungulates, and how they might be interacting with pasture use, with some idea of how that might imprint on snow leopards or predators in general. Some more context from literature, I found it very interesting to know. I mean, this is an old statistics, and I'm sure that it has been updated since. But roughly about a third of the global land surface is grazed by some sort of livestock grazing. And interestingly, and I'm sure it's more now, 752 million people who are completely dependent on livestock grazing live on less than $2 a day. So you can imagine how important livestock grazing is in the context of one's livelihood across a huge land surface as well. And in all of this, it's not surprising that human-wildlife proximity, especially when we talk about ungulates, because ungulates are sharing resources with, well, wild ungulates anyways, are sharing resources like grazing areas, watering grounds with these livestock. So this proximity is really there, especially for ungulate species. And interestingly, and a lot of us probably have read a lot about competition, resource competition, exploitative competition, especially in our landscapes in the high Himalayas or Central Asia. And a lot has been written about it. We have written very little, or there hasn't been much work done on the potential of disease transmission. But all of these things that I've just spoken about really provide a platform for this to occur. I think that's where my sort of, intrigue in looking at some of these aspects of disease transmission really came in. So why is disease important? Especially in the context that I just said, diseases can impact agricultural economics of herders, particularly, those 752 million people that live on less than $2 a day. Imagine if their livestock was impacted by disease. That is a huge sort of livelihood impact that we're talking about. But they're not only important for agriculture economics, but they can be also important for wildlife conservation, because diseases can impact wild populations in many, many different ways. And disease is very broad, and it's a very general term. Within that, I specifically looked at gastro-intestinal nematodes. These are essentially endoparasites or worms in the gut. People have them, wildlife has them, livestock have them. All of them have these, parasites to varying level in their, internal sort of systems. And gastrointestinal nematodes are not a disease per se, but they're just one form of parasite. And I was very intrigued to study them for three particular reasons. One was they can be key determinants of fitness. There's enough studies done in wild ungulates and domestic ungulates to show that varying levels and times when species have GINs, which is just a short form of gastrointestinal nematodes, they can impact things like growth rates. They can impact things like the amount of milk being produced by females. They can impact even fertility rates of both wild and domestic ungulates. So they are a key determinant of fitness. And I think that intrigued me more, because then that aligns a lot with different narratives around, if pastures are degrading, they are also impacting fitness. So I didn't want to necessarily look at just diseases as a singular, sort of issue in these systems. I wanted it to be more holistic, and I thought that's where GINs are a bit more of a better sort of disease to study than, let's say, a very specific disease that might result in a huge amount of mortality. GINs also result in susceptibility to other diseases. In other words, let's say if you have high GIN loads, you might just become weak. You might not be able to ingest food and digest it the way you would normally do, which will then potentially lead in the acquiring of other diseases, which might then be fatal. So I thought they are a gateway to this sort of impact of health So I think that was the other reason. And lastly, and I think this is very important in our systems, especially, that a lot of their life history, which we'll speak about in a bit, is governed by climatic factors. So with changes in climate, you might have changes in the way gastrointestinal nematodes fluctuate across the year. So I did this study particularly in Kibber village. As I said, one of the co-authors of the study is here, Rashmi as well. She did a lot of the groundwork for this, so I really, really want to thank her, especially today. So yeah, a lot of it was done in Kibber village. This is what it looks like, just for your reference. It's this high altitude plateau at around 4,200 meters above sea level. As you can see in the distance, that's the village. And these are the grazing grounds around the village where livestock graze as well, and wild ungulates, particularly blue sheep, are. So just to give you a sense, very arid landscape, high altitude landscape, very seasonal landscape as well. With high winter snows and very cold temperatures and quite dry summers as well. So just to give you a context of what really happens when we're talking about parasite transmission, right? So if you just follow the sort of cursor on the screen here, in these landscapes, because these are multi-use, there are both the wild ungulates, the blue sheep, and the domestic ungulates, in our case, small bodied livestock like sheep and goat. Essentially, the adult parasites, the adult GINs, are in the body, maybe in some form, in the adult form rather, in the gut. And when they reproduce, they form eggs. The eggs come out of the host, both from the wild host and the domestic host, into the feces, right? And this is deposited onto pasture, which is co-grazed by these two hosts. When they come out into the feces as eggs, depending on the abiotic conditions, primarily the temperature and how wet it is, the precipitation, these eggs then hatch and survive or die, depending on the conditions, as I said. And then they make this journey from egg to L1, which is larva 1, larva 2, and then larva stage 3. These are just different stages, going from, let's say, a kid to a young one to an adult to finally a mature adult. And at larva stage 3, they either stay at the bottom in the soil, let's say, of the ground, or they migrate upwards onto the stalk of these vegetations. And that larva stage 3 is the infective stage. Essentially, when, let's say, a blue sheep comes or a livestock comes and grazes this, whatever this may be, a stiper, for instance, that they both eat, they can then ingest the infective larva. And then that infective larva becomes an adult, which reproduces, and then the cycle continues. So you can already imagine if, let's say, something deposited by the sheep or the goat in the pasture becomes L3, and for whatever reason comes onto this stalk, which is then eaten by the blue sheep, that's where the transmission loop occurs, and then it continues, so on and so forth. So yeah, that is just to show you conceptually how the life cycle of these parasites work, which we also modeled, and I'll talk about that in a second. So what were the aims of the study? It was really to understand how the system, in this case, the Kibber landscape, is behaving in terms of parasite transmission, and also look at potential solutions. Why is this important? Because then we can use this to have time and space specific interventions that align livestock growth with wildlife conservation as well. How did we do this? We use multiple methods. We try to understand where hosts are found, so their distribution and their density, because both of these will impact the amount of parasites they can shed onto pasture and then contribute, as potential infection. We also importantly looked at social factors that might be in play in this landscape to manage livestock. For instance, if people are already knowing which pastures might be contaminated with disease and not, or disease per se, but with parasites, and not take their livestock there. Not accounting for that in our study would just not be the reality then. So we talked a lot to the livestock herders that know a lot about all of this to understand if anything of that was in play. Then, of course, we looked at actual endoparasites in our hosts. This was all done in the field by collecting fresh fecal samples of both the blue sheep and the livestock and analyzing them. I won't get into the methods, but we can discuss it if that's useful. And then eventually, based on all these 3 points, we used a parasite transmission model. Again, we can talk more about it in the discussion, to actually model the pasture infectivity, or in other words, to see when are the times in the year when pasture infectivity increases and who might be contributing to it more, the blue sheep or the livestock. So very quick points about host distribution. Just to situate ourselves again, we're here in Indore village, which is in the Trans-Himalayan region of Himachal Pradesh. And by doing a lot of work with herders, but also doing field surveys, we delimited this, sort of timber livestock pasture area, essentially. And this is where the livestock goes through the year. And we did multiple field surveys to also see where the blue sheep are. And what we realized is blue sheep also use the same area through the year. Obviously, there are some variations as to where they go, and there's some seasonal movement, but very, very limited. As you can see, this area was not really used by blue sheep at all. And that's because these are really high glaciers that, in fact, even the livestock don't use, but it still comes under the jurisdiction of Kibber village. So essentially, it's this area that both the livestock and the blue sheep are using year in, year out, and having, sort of pasture, what's called co-grazing of pasture. We also found that blue sheep were at much lower densities, at about 5 blue sheep per square kilometers than sheep and goat, which were at around 22. So just keep that in mind in terms of the volume of feces coming out of each host and potentially how much parasites they might be contributing to this co-grazed pasture around Kimber. Then we, to understand the social factors to manage livestock, we did about 32 interviews with different households. These are households that all own livestock, particularly a small sheep and goat. They also herd them. There's a system where they change herders every day, I think. So everybody has a chance from these 32 households to go out to herd. And we found no explicit disease governance grazing patterns. Or in other words, people were going out to graze, not thinking about which areas within that Kibber landscape might have higher pasture contamination. They were going out more from the perspective of which might be the best graze for them. So that was interesting. There was no natural or synthetic use of anthelmintics. Anthelmintics are essentially anti-parasitic drugs. They could be natural, such as Artemisia sometimes, or synthetic, such as something that you buy off the counter. So none of that was being used. And all of them agreed that winter survival is super hard. These are cold landscapes. But livestock still goes out in the winter to graze. There's very limited fodder available, at least small-bodied livestock, if possible. But there was many different results here, and those are all in the paper. But one of them I wanted to highlight. This was basically, we asked them, if you had any diseases that were impacting your livestock and things like that. And what you interestingly see is that a lot of people, the most cited and these are sample sizes here at the bottom and percentage on the y-axis. The most cited quote-unquote disease/issue was ectoparasites. So those are definitely something to think about. And especially in these cold areas, ectoparasites, particularly in the winter, tend to be an issue. But that was not necessarily our focus for this work, of course. But interestingly, when you go down the sort of bar, you see things like nasal discharge, coughing, diarrhea, swelling of the throat, and even red eyes popping up. And interestingly, while these are not diseases per se and most symptoms really, all of them tend to be symptoms of parasitism to a certain level. I mean, you could be coughing because of, let's say, a disease like CCPP, which is present in these areas, but it's very likely that these are also in part there because of some sort of, GI and parasitism, which maybe people are not mapping because you don't necessarily see these parasites very visibly. So I just thought this was an interesting result that we got. So moving swiftly on, we then looked at endoparasites in the host using the flow tag method. And we basically were looking out for particularly these strongyle eggs or strongylus species. There's many different types of parasites, endoparasites particularly. But we were focusing on strongylus for two reasons. One is there are different species of strongylus in particularly ungulates. They're known to be physically detrimental, fitness-wise detrimental, and also particularly some species like Teldosasia, they're found in these cold climates as well. And the transmission model that we had was based on the life history of the strongylus eggs as well. So just some background data, we had about 124 samples from 30 unique days. And of these 124, at least 58 had these strongylus eggs. Blue sheep, we had about 116 samples from 38 unique days. And I'll explain why unique days are important in a bit. And 62 of them had strongylus eggs. So already from this very basic analysis, we see that there is a possibility of transmission. We know these hosts share pasture. We know there isn't anything explicitly to limit their transmission, and we also know that both of them do have strongless eggs. But is what's actually happening in terms of transmission? We use this data to model that. This is just a graph to show how eggs. So this is eggs coming out from each host are coming onto pasture. There was very little eggs coming out from either host during the early parts of the year, with some variation, of course. And it was through the middle and the latter part of the summer that eggs were really heightened for blue sheep and sort of similar for livestock. But it doesn't matter too much how many eggs are coming out from hosts, because if those eggs are not making that journey, which I showed you in that conceptual model, from egg to L3 to become infections, if they're not making that journey, if it's too cold or whatever, then these eggs don't make much sense. So these eggs coming out were actually the input into our model, really. So just to give you some context. So then we went ahead and modeled pasture infectivity over time. Lots going on in this graph, but I'll sort of walk you through A, B, C, D, and E. Essentially, A, what it shows is, and for all of these, the x-axis, a number of days going from 1 to 365. 1 being the 1st of January for the year 2018. We did this, if I'm not mistaken, going to 31st December, which is the 365th day of 2018. And on the y-axis, we have different y-axes. So let's look at graph A. For graph A, it's L3 on herbage, or in other words, how much infection the L3, the infective larvae, which go back into the host, is present on pasture on that given day. So as you can see, nothing much is happening for the start of the year, nothing much, and it's around this 100 and just before to the second 100th day of the year, around the 6th month of the year, where you really have this spike in livestock and in wild ungulates, in this case, the blue sheep. The contribution from the livestock is much higher than the ungulates, as you can see from this test that we have, which is significant. So in other words, livestock in our systems are contributing more, significantly more to pasture infectivity than the blue sheep. Panel B is essentially just combining the two results together, essentially, of hosts in the system, if you just want to see pasture infectivity across. Panel C basically shows when the fluctuations start. So as I said, nothing happens for a long time in the year because it's cold, and then the fluctuations start mid to late summer, and then they die down again from autumn onwards. And interestingly, to explain why the system is happening the way it is, I think graphs D and E explain a lot. D is the precipitation data, daily precipitation data in millimeters, and E is the temperature data in degrees Celsius. And what essentially happens, if you look at the temperature, it's around day 130, 140, when the temperature increases above 6.5, that's when you start getting very minimal. It's not being able to be visually seen here because of the scale. That's when some activity in the L3s happens. And it takes a little bit of time, almost those 2 months, till you have this build-up and sort of peak in pasture infectivity. So it's a lot to do with warming temperatures post the winter. And then you have this warm period, which then goes back down post the autumn. And that's when the infectivity also goes down. And interestingly, in this warm period, sort of between, let's say, May, June, July, August, and early September in our systems, is also a bit wet. That's when most of the rain happens. And then, towards the winter, it's actually super cold, but it's not that wet. I mean, you might have a lot of snow, but that's very, very limited as well to a certain days. So that's very interesting. And this is classical slow burn system dynamics of a parasite. What that means is, not much happens for a long time. Then the conditions in terms of temperature and precipitation becomes good. Then it needs this buildup slowly of, different infection on the pasture to then reach its peak, which then dies down. So this was very informative as to when, pasture levels are facing highest infection rates and what might be done about it. So then we asked a little bit more to see which climatic cue might be, more important to know for the herders that infection might rise. So there is this quotient that we developed, which basically asks to which extent, to what extent does precipitation drive transmission over, let's say, temperature. And essentially what we did was we looked at the infection larva's area under the curve using actual precipitation data divided by area under the curve of high precipitation data, or in other words, if precipitation was not a limiting factor. And essentially we conceptualize it to be the following: if this quotient is close to 1, then this is more a temperature-driven system. Temperature is more the limiting factor. If the quotient is close to 0, then the system is more a precipitation-driven system. And our quotient was much more closer to 1. And in other words, it's a more temperature-driven system. But the way to understand it is not to say, oh, your rainfall is not important. In fact, it is. It's just to say that the temperature is a better cue to understand transmission. Or in other words, if a herder has, let's say, an understanding as to when temperatures might cross that 6.5 to 7 degree threshold during the day, average temperatures, then that's when the buildup of infection might happen. And then, as you could already see from the previous slide, that maybe a month or two down from that increase in temperature to 7 an average a day, that's when the peaks will come. We also looked at how this might change or might have changed in the last 20 years by looking at historical data. And again, what we find, and there's lots going on in this graph, is that the hottest day of the year for these last 20 days lags by 12 days from the peak of infection. Again, I think what I'm trying to show here is that in our system, it appears that the temperature is a very good cue to know when peaks of infection might happen. And if you know when peaks of infection might happen, you can already start sort of doing something about those to reduce them. Just to allude, this is sort of the last section of the talk, that these are multi-use systems. So both livestock and wildlife, work and impact each other, as we already saw from some of the disease transmission information. But it's much harder to do anything about all of this in wild ungulates. And it's arguably a little bit easier to do or manipulate things in the livestock. So at the back of all this knowledge that we generated about how parasites infect seasonally, could we do something to better align livestock health with wildlife health? We didn't do it on ground, actually, but we did sort of a scenario analysis through these 4 hypothetical theoretical interventions. And we wanted to do this first to understand what might happen if these interventions were done, and then talk to our stakeholders to see, okay, is this something you think is important, needed at all, and will this work? So the 4 hypothetical interventions were essentially had 2 elements to them: either housing livestock so they don't infect, put those eggs on pasture, which then become infected larvae, so they're kept away from pasture for a certain time, or you keep grazing them on these pasture, but you treat them at certain times, with these anti-parasitic drugs, so then they don't infect pasture. Essentially, the way it's done in the model is you switch off the egg input from the livestock for that given period of time to see what might happen to the output of the infection on pasture. So if you remember this conceptual model again, what essentially we're then doing is cutting this egg input onto the pasture from livestock for whatever given time. And the 4 scenarios were, because we had this very well-defined peak that lagged with temperature, we said, okay, what if we house them or treat them for 2 weeks? Because, we can't do this for the entire year. These are resource-limited systems as well. And you don't want to create any anti-helminthic resistance as well, right? Like if you keep giving them drugs for the entire year. So let's do it for 2 weeks prior. Let's do it for 4 weeks prior, because maybe 2 weeks is too short. Let's do it for a month prior to the peak, because as you remember, these are slow burn systems. So it takes some time for that sort of, peak to be reached, even though it starts almost 60 days before, the infection buildup starts 60 days before, albeit it's very slow. And maybe even 1 month is too short. So let's try for 2 months, because prior to the 2 months, it's anyways too cold. Prior to 2 months of August, which is when the peak is, it's kind of cold till April in our systems. So we did that. And essentially, we ran these models and we looked at the effectiveness by calculating the area under the curve. So the area under the curve, normal as it was called, had about, a total, and this is the summation of the infection, was about 14,201. And the first intervention, which was, treating for just 2 weeks, didn't really change much, reduced a bit. 4 weeks didn't really change much, reduced a bit. Even 1 month didn't really change much. But the 2 months prior treatment actually reduced infection by 80%. So as you can see, and this aligns very well with the whole slow burn system, is it doesn't matter if you don't start treating at the time when that slow buildup happens, which is not very visual because maybe you don't have the impact of those very, very minimal larva, infective larva on your body. But if you don't start intervening then, it's too late. It doesn't matter much if you start treating close to the peak because already the buildup is too high potentially. But as you can see from this result, if you target the intervention at the right time, you can almost negate the impacts of these parasites almost altogether, at least by 80% in our theoretical intervention. And as I said, yeah, this is consistent with the slow burn system. So yeah, just sort of summarizing all the key points, endoparasites seem to be a blind spot in our system. Especially, I think it's important, even though they might be occurring at low numbers, a lot of our systems tend to be overstocked. I mean, we have to really discuss what that might mean in terms of overstocking. But my point being that they're present, they might be impacting livestock health and potentially even wild ungulate health, and they're not visible. So they tend to be a sort of a blind spot, especially as you remember from that data as well. When we asked people what's impacting your livestock, they were saying symptoms of endoparasites, but not necessarily calling it that. Spillover from livestock is very likely, at least in our system. As you can see, they almost contribute to twice as much partial infectivity as ungulates. These are slow burn systems. Moderate temperature and slow evapotranspiration results in this sort of trend of historical pasture use or egg shedding. Then accumulation of larva, infective larva on pasture, which then governs this infection potential. So what does all of this really mean? I think it's important to contextualize whatever we've done, both from a social and an ecological point of view. One way to do it is by integrating a lot of the knowledge that the herders were giving us with our model outputs. After we did all this scenario analysis around the interventions, we went back and spoke to the herders, especially the 32 herders, to be like, okay, this is what we found, and this is what it looks like. And even though intervention 4 seems to be super effective, it can be quite unrealistic and socially unacceptable in our landscapes. Housing for 2 months at a critical period of forage, because those are the lush summer months, short periods, would mean almost, devoiding the livestock of the feed that makes them survive in the harsh winter sometimes. And there's anyways shortage of fodder, so you don't want to enclose them and just give them fodder. Peak summer forage is valuable. Our landscapes where we work are primarily Buddhist culture and religion, and there's this concept of right to live where you don't want to cage something, so you let it be out in its sort of natural habitat, in this case of the livestock, the pastures. So housing really wasn't sort of something that was realistic or socially acceptable. But based on our outputs, we did get an understanding of the transmission seasonality and also the magnitude, if you remember. So that the herders were very keen on, and they were excited to see that result. And what we sort of spoke to them about is how we can use that output from these global method models. They're just called global method models, the parasite models that I showed. We can then use that seasonality and detect unhealthy livestock. There are ways to know which particular individual within a population might be having more endoparasites because they might have certain symptoms. There's a 5-point check that you check 5 points. If the eyes are just that the eyes are less red, they're more sort of whitish in color. Is there a bottled jaw? Things like that are signs of parasitism. So if you continually, and these herders are with the livestock day in and day out. So if you already look for signs of parasitism around that 2-month period before the peak that can be predicted for that given year, you can already start treating those individual livestock with either natural anti-helminthics and or synthetic and anti-helminthic. And I think that's important to note because rather than treating the entire herd, which can then build resistance, you're actually treating just individuals that A might be contributing more to partial infectivity, and then B might be impacted by these parasites themselves. So there's a lot that could be done, at the back of this study. So that was very, very encouraging. And this is also a cost-effective way to potentially use the outputs of our study in our context. Some caveats, sort of last slide. I think it's very important to ask the questions, even though we sort of model the GIN transmission and whatnot, are they actually at physiologically detrimental levels in our landscape? Because sometimes you can have lots of GINs, but they might be dealt with by populations in different ways, or sometimes you might have very low loads, but they're still very detrimental. And it's very hard to actually map that physiology impact onto the individual and population. But there are means to do that. There's some really interesting eco-immunology work being done there. Even though we have a lot of the system understanding now, it's actually hard to translate that into stakeholder action, in our case, primarily the herder. So we've been sort of seeing what to do there. And at the same time, while we understand there is this annual overlap between the wild and domestic ungulates, sometimes this very, very fine scale contact patterns, which we might only get from collaring data, for instance, which actually lead to parasite transmission. So I think it'll be interesting to see how we can slice and dice these contact patterns across scales. And interestingly, and I think this is a bit of a wild one, to see if at all predators like the snow leopards, which might impact the movement of the prey, particularly the wild prey, do they play a role at all in determining this transmission? There's some interesting work done on tigers and spotted deer in this context. So I think that's an interesting one to look at in the future. Just to finish off, I wanted to acknowledge my field team. I mean, there's all the people from the Kibber village who helped us do not only this study, but many, many other studies. They've been working for over 15 years in these landscapes in some of the harshest conditions. So I really, really salute them. I mean, they are truly the reason why we get to do whatever we do. So yeah, just a shout out to them. My supervisors, EJ Miller-Gullen, she was at Oxford, really pushed me to think a lot about the whole socio-ecological context of disease transmission. Dr. Eric Morgan, who's a specialist in parasitology, and Dr. Kulbhushan Singh Suryavanshi, he does a lot of, or rather has done a lot of work in the Trans-Himalayan landscapes of India. So it was really lovely to have them as a sort of super team to work with. And finally, just the herders and the people of Kibber village who are so warm and welcoming. And just a photo here to show, that these guys are out grazing their livestock day in and day out, regardless of weather and season, time of year and whatnot. So it's really commendable that they live the lives they do. And I just hope that through my work, I work with them and co-design interventions with them that is helpful both for their livelihoods and the wildlife that lives around. So with that, I'll stop. I hope I've not taken too much time. But yeah.
Thank you so much, Munib. That was fascinating. Thanks for sharing your study about how, using these socio-ecological lenses can help us understand disease transmission. And so lots of questions are trickling in, and while they start getting collected, I thought I'll get your thoughts on some questions that I had. So I was curious about how,
Sure.
I was curious about how when you model these different hosts in your system, how do the dynamics change when you have migrating hosts?
Yeah.
So would you develop a completely different model?
Yeah, that's a great question. I think, and that was the follow-up study actually from this, where we tried to model, this partial infectivity in a migratory system where the livestock are migrating up from the plains in India up to the mountains, and the wild ungulates. In that case, it was the ibex, are sedentary. And we had another case study from Kazakhstan where the wild ungulates are moving and the livestock are sedentary, just to look at how movement impacts all of this. The way we did it, which is just one of many ways, is to sort of then incorporate the movement spatially, in a spatially explicit manner. So essentially think of a map in your mind and this cycle playing out along that migratory route of that individual or that population. We looked at populations, we didn't look at individuals. So let's say for the livestock,
Right.
we looked at egg output at a given space, and we compartmentalized it. We said, okay, we will look at it in their winter, spring and summer distributions, because then the spring is the same as autumn. So they do these movements up and down. And then we had multiple, global method models being generated for those landscapes for the times they were there. So that's one way of doing it, which is a bit of a crude way. I mean, perhaps a more elegant way is to then link these cycles as they're moving rather than in these compartments. So yeah, that's a couple of ways that I can think of. What's interesting there is,
Right.
because there is a lag between deposition and then, becoming L3, at least that's useful to know, because if you deposit eggs, let's say in landscape A, then it's impossible that at least those endoparasites will, be infective in landscape B because they're deposited in landscape A. So you can get a story of, these seasonal.
Yeah.
So yeah.
Yeah, and once you can build that model, you can also then do a similar intervention scenario simulation, and then I think it will be very different, the kinds of interventions that you'd want to do. Yeah.
Yeah,
Thanks.
absolutely.
Thank you. Yeah.
No problem.
I also wanted to ask you about what existing surveillance and monitoring systems are there in your study sites, and also
Sure.
from your experience globally in these mountain ecosystems. What systems are currently available?
So I know, for instance, at least in our sites in northwestern Himalayas, there are departments like the sheep husbandry department or the animal husbandry department that sort of look at the diseases or health of livestock particularly. And then there is the forest or the wildlife department that is more concerned about the health of the wildlife. There are basic diseases that are on the radar because they might be national priority. Things like mouth disease, which is a virus, even PPR, which is a virus again. These things are sort of more in the limelight because they're known to be very, very detrimental. They result in huge losses. So there are vaccination drives, for instance, at least for livestock, around those diseases. And I think that's predominantly true for the larger Central and South Asian landscapes, at least the mountain landscapes. And I think that's where the gap for parasites come in, because they might not be that visible, or they might not have that, impact that results in the death of livestock, for instance, the way PPR might have. So I think those, have seen very limited interventions or even studies around that. But yeah, I think there's a very good amount of intention and intent of these different departments. I think sometimes it's, at least in our sites, it's a little sad because they don't necessarily communicate. Like the livestock departments might not necessarily communicate with the wildlife departments, even though they're all so interlinked in terms of transmission. So I think that's a bit of a gap.
Yeah.
Sadly. Yeah.
All right. Thank you for sharing so much with us today. So we have a few questions.
Sure.
Yeah. So I'll just read it out for you. So Hussein Ali is asking how the infectivity of pastures was checked from January through June.
So that's a good question, Hussein. So two answers to that. So remember, pasture infectivity is a function of the model output. Whereas because it's the eggs going from being eggs to L3, which is entirely modeled, right? It's the egg input that we actually checked on pasture, which, as you saw from one of the slides, the egg input slides, we had some data from January. In fact, I think we collected egg data from pasture throughout the year. One reason is because Kibber village is so high up, you're already at this 4,200 meter landscape, and the livestock don't go out too much. So even though there's a lot of snow, these are still rolling, predominantly accessible pastures, although not all areas are accessible. So I think partly because we could have that access and the luxury of going out to pasture to look at, where the eggs were being deposited because the herders were going. And to answer your question about actually infectivity, that was entirely modeled. So then that from the egg input, which is a field data, we then incorporated life history parameters of that particular species of parasite, and then incorporated also abiotic conditions such as temperature and precipitation to look at what the infectivity is like.
Yeah.
Yeah.
So Hussein, maybe I'll move to the next question.
Sure.
Yeah, next question is by Aliana Norris. She asks,
Right.
they ask, did you find any evidence for presence of other types of nematodes or endoparasites that
Mhm.
might have a different life cycle or additional hosts
Yeah.
from strongyles,
Yeah.
such as Echinococcus or others that might also threaten human carnivore health?
Yeah, that's a great question, Ariana. So thanks for asking that. So a couple of answers. There's always two answers to a question, I feel. One is the endoparasite species slash groups that we were able to identify was quite a function of the method being used. So the one that I didn't really explain it much, but I used the flow tag method and then the microscopic sort of investigation of those floating eggs. So that's very specific to certain types of endoparasites. To get a sort of a whole range of what might be there's definitely other methods that one could use. So I wouldn't necessarily look at my study as an inventory of potential endoparasites in our hosts. The method used was intentionally used to be robust for the strongyle nematodes, and that was for a couple of reasons. One, because as I said, the strongyles were the input for the model that we had developed, but also there's enough evidence in literature to show that strongyles are, quite detrimental, at least to ungulate hosts. Echinococcus, I think, is a huge issue, particularly in canines, if I'm not mistaken, in canid species, if I'm not mistaken. Please correct me if I'm wrong. So we didn't have those come up because just of the way we sampled. I know, for instance, in our migratory case study, which Manvi asked about, there again, there are some studies done in the same landscape that get things like liver fluke, which is Fasciola species, for instance. But we never got liver fluke in our samples because the way to actually, sample for liver fluke is completely different. They don't float in the saline solution the way we did it for strongyl eggs. So that's what I would answer for your question. I think there's certainly a lot more endoparasites out there, particularly as also I keep talking about these co-grazed areas. It's not only about the ungulates, right? It's also that they co-grazing this with marmots. Well, not many marmots here, but mostly pikas, maybe some birds. So there might be some cross-transmission. Although, at least for parasites, there's a very strong taxonomic link between transmissibility. Although, yeah, so just a couple of points on that.
Thanks, Munib. Yeah, Liana, I'll move on to the next question then. There's another question from Hussein. One solution you shared was cutting the transmission cycle, but in many cases, this is not. Did you check the infectivity of pastures from livestock with vaccinated livestock?
So, interesting question, Hussein. So, I think what I would like to allude to firstly is because we were studying parasites, it's not a case of vaccination. It's more a case of use or non-use of anti-parasitic drugs. Because vaccination is more sort of being very specific about, let's say, disease X, vaccinating livestock against, let's say, foot and mouth disease, which is a virus or some bacteria. So in the case of parasites, there is no vaccination per se, but there is a treatment or no treatment, which is where we had those scenarios. But if we were to take the point about vaccination, I think that's a very interesting point because let's say if there's disease X in our systems, PPR is very prevalent in our systems, and even CCPP, which is a pulmonary pneumonia, a virus-based pneumonia. From my understanding, at least in the Indian Trans-Himalayas, there is no vaccination for CCPP, but there is for FMD virus, for instance. So if you have vaccinated livestock with the right efficacy and right dosage, then the transmission loop is completely cut. And that's what a good vaccination program does, for instance, with COVID being a great example in humans, for instance. But in terms of CCPP, where there is no vaccination, you might actually do other things to reduce, the potential of a host getting impacted by CCPP, or you might, give it good feed or things like that, or keep it away from pasture where CCPP is there because you don't have any vaccination. So there, the transmission loop is not cut by the treatment; it's cut maybe by social interventions of where you take the livestock or you might feed them certain things so they don't get impacted by that disease. But yeah, I would like to make the distinction that in our case, it's not about vaccination. It's more about treatment through anti-parasitic drugs, if at all, or the way you graze these areas. Yeah, I hope that's clear. Yeah.
Yeah, thanks, Munib. The next question is from Bonnie. They thank you for the excellent presentation, and they ask, how does the vegetation affect parasite transmission? For instance, do the L3 larvae show a preference for certain plant species? And if so, how is plant composition impacted by the temperature and precipitation patterns you described, and or grazing of both shorts and Bharal?
Yeah, that's a great question. I'm really enjoying thinking through these. But these are all bits and pieces of caveats in the study, so it's great to have them be highlighted. So yeah, I think we know very little about how vegetation and these parasites interact, at least in our landscapes. But what's important to note is to get that infectivity of L3, the infection larva on herbage, we essentially want to understand how much herbage there is. Like, or in other words, imagine if the landscape was a complete desert with no herbage, then there would essentially be no transmission because all the parasites, at least the ones we are concerned about, would be, let's say, in the soil itself. So there needs to be some herbage for then the parasites to come up from the soil on top, which then gets eaten. And that in reality is a function of what kind of herbage is out there. For instance, if you might have carrageenan, which is a type of plant in our system, which is barely eaten by either livestock or blue sheep, because it has these nasty thorns. Now imagine if a pasture is completely full of carrageenan, even though the the L3 come up onto the, these tips of the carrageenan, they will never be ingested by the host because they don't eat them. In other, or in contrast, if it's full of, let's say, stipa, which is a grass species, if I'm not mistaken, and it's full of stipa, let's say there's no other species but stipa on that pasture, then it is highly likely that, that L3 will migrate from the soil up onto the top of stipa, and then there's transmission all around because even the blue sheep is eating it and the livestock are eating it as well. So it really does impact pasture infectivity and then transmission. The way we modeled it was to just take a sense of what the biomass of these places are in terms of vegetation in that period of time, because there was some studies done already to show, what the average biomass of a kibber pasture is in the summer, how much is it in the winter, and how much is it in spring. And we just use that average to say, this is the amount that can be transmitted from A to B. That's very crude, of course, because that's assuming that all of that biomass can be eaten by the hosts, which is not true. So the best way to look at our results is to take them with a pinch of salt and say, that's the maximum level of pasture infectivity that can be there. Because, we're assuming that whatever is out there can be edible by the host, which in reality is not true. It's a function of actually what the vegetation composition is. And one can incorporate that in the model by A, looking at actual biomass of vegetation and composition, and B, looking at dietary preferences of hosts. So yeah, that's, I think, something to look at in future studies as well. So, yeah.
Yeah, thanks, Munib. Bonnie, I'll move on to the next question, which is by Aliana. They ask, how do you think the possibility of climate change, increased temperatures, or longer warm seasons might impact the transmission? Possibility of longer or more intense infection seasons, maybe?
Yeah, that's a great question. I mean, this is something that I looked at a little bit in a case study with Saiga antelopes, and that's published as well. I think the answer is not linear, because climate change itself in our landscape is not necessarily linear. What I mean by that is, yes, one would expect warmer temperatures to be more conducive for parasite growth because, like -40 is not great for any life form out there in the winter. But it's not only about increasing temperatures, because we also need to have enough, precipitation to compensate for that, increase in temperature. Because a lot of times A lot of parasites have developed life history patterns to, finish their life cycle based on, let's say, these narrow temperature and precipitation regimes. To give you a sense of, or an example rather, so there is Humongous contortus, which is a parasite species found predominantly in wetter areas of the world. So they really don't care about how warm or cold it is because, generally in the tropics, the temperature band is very narrow in terms of how cold it gets or how warm it gets. You really have these seasons of rain coming in and coming out, like monsoon, for instance. So they really bank on the rain for their life history. Alternatively, a lot of the Arctic-based parasites, they are known to utilize these short periods of warming summer, similar to what we see in our landscapes. Because it warms up and there's a little bit of rain, then it's fine. So what I would predict is, yes, with warming temperatures, you might increase pasture infectivity. I don't think it'll change the seasonality of the infection. It might change the intensity, as you said, but it would very much also be a function of availability of precipitation in that area. You might have, might need that equivalent amount of increase or decrease. And I think there's a mismatch in some areas in the mountains where you're getting a lot more warmer but drier summers. So then it almost compensates. The increase in heat is compensated by the decrease in precipitation. So I think I would look at it a bit more holistically in terms of the abiotic conditions. But yeah, I think the prediction would be what you suggested. Anyway.
Yeah. So just to add to that, it might be there might be some work done on what traits of parasites are more vulnerable, or or they're more likely to spread. Some trait-based analyses might have been done. It's, something interesting to check or look for. Yeah.
Absolutely. In fact, just to add to that, if people are interested, there's lots of interesting work done on exactly that by Susan Kutz's group. They're based in, I can just type in the chat. They're based in Calgary, if I'm not mistaken, in Canada. So they do a lot of this up in the Arctic, which I think is a very comparable system to a lot of our mountain systems. So I would really recommend reading their work. I think they've done quite a lot of great work.
Thanks, Munib. We're nearing the end of our webinar. There's just one last question, and then maybe we can call it a wrap. It's by Bilal Mustafa. They ask, they thank you for the amazing presentation, and they want to ask, did the transmission and disease vary across different sheep-goat breeds?
Yeah, nice to see you, Bilal. Bilal is a good friend from across the border, so it's nice to have familiar faces. So the answer to your question in the context of our study is we don't know, because we actually didn't look at breeds as a function of, pasture infectivity and things like that. I think it's a very interesting one to look at, both from a parasite's point of view, but also generally in diseases point of view, because different breeds can have different health status. So I think it would be interesting to look at that. In our case, I think we have very specific breeds in Khyber, especially from what I know of sheep and goat. I don't think there's huge variation in breeds, so I would not be super surprised if there isn't a breed-related impact on pasture infectivity, at least in our case study site. But if you go into, let's say, Changthan, which is eastern part of Ladakh, just north of our study site, there you have different breeds. You have the Changra goat, which gives you Pashmina, or Kashmir as we call it as well. And then you have like a different breed of goat. And then you have like, I think 2-3 breeds of sheep. One is more for meat and I think the other is more for wool. So I think, it'll be interesting to look at what trade-offs there might exist in those breeds. I haven't really thought about it much in terms of, which breed might be more resilient to the other. I mean, I would imagine a breed that's more migratory might have these evolutionary adaptations to different parasitisms, because they've been migrating for generations, over, let's say, a breed that is more trans-Himalayan, like the Changra goat. I think once you take the Changra goat down into more better habitats, I think she might not, or he might not be, they might not be that happy, for instance, just to experience that higher level of humidity and temperature related parasitism. So just a couple of thoughts on the breed.
Thank you. So let's thank Munib again for a very insightful session. Thank you so much, and thanks everyone for joining us today. We'll see you in the next webinar.
Yeah, thank you so much, Munib, and thanks, Manvi, for giving us your time. And thank you to all our participants. And we look forward to seeing all of you all again soon.